The protocol demonstrates a rapid method for identification of bacterial proteins using antibiotic induction and mass spectrometry. The main advantage of the technique is its speed and simplicity. Sample preparation is uncomplicated. The technique can be extended to the characterization of any pathogenic bacteria, such as virulence factors or antibiotic resistance, to better inform appropriate treatment for a bacterial infection.
To begin, use a sterile 1 microliter loop to harvest bacteria from single colonies grown on LB agar plate. And transfer to a 2 milliliter O-ring lined screw cap microcentrifuge tube containing 300 microliters of HPLC grade water. Then briefly vortex the tube and pellet the cells by centrifugation. Add a 0.75 microliter aliquot of the sample supernatant on the stainless steel MALDI target and allow it to dry. Overlay the dried sample spot with 0.75 microliters of a saturated solution of synapinic acid prepared in 33% acetonitrile, 67% water, and 0.2% trifluoroacetic acid, and allow the spot to dry. Once the spot is dried, load the target in the mass spectrometer. Open the acquisition software and click MS Linear Mode Acquisition. For mass to charge range, enter mass to charge of the lower and upper bounds into their respective fields. Click on the sample spot to be analyzed on the MALDI target template. Then depress the left mouse button and drag the cursor over the sample spot to specify the rectangular